Hello and welcome everyone to this next podcast on eukaryotic gene regulation. On the last podcast, we talked about the general ways that eukaryotes can regulate their genes. In this podcast, I want to focus in on regulation at the level of the chromosome and chromatin, and at that direct level of transcription, meaning talking about those transcription factors that bind to the DNA. This first one is just going to talk about how we make that chromatin accessible for these transcription factors. So it's appropriate we start talking about chromosomes and chromatin first, and then we'll move into transcription factors and how they directly help turn genes on and off. Okay, so let's talk about chromosomes first. And to do that, I'm going to draw a cell here with its nucleus right here. Now, when I first learned about chromosomes in the nucleus, I learned that they were just all kind of crammed in there. But what we've learned is that chromosomes occupy distinct areas in the nucleus. These regions that each of these chromosomes occupy, and I'll just kind of rope off a couple of them, these are called chromosome territories. Now, another term I want to talk about is called transcription factories. And what these are regions. I'm just going to circle a region right here that doesn't have any chromosomes in it, but will contain all the transcriptional proteins necessary. So in these transcription factories, there will exist RNA polymerases and transcription factors, as well as other proteins necessary to turn genes on and off. And there will be several of these transcription factories in a cell, but the one I drew here may pull in DNA necessary that's containing genes that need to be transcribed into this region. It'll unravel these local pieces of DNA, and that way they're concentrated in areas where there's a high concentration of these RNA polymerases and transcription factories. In this way, we can effectively turn on a whole set of genes without having to move around RNA polymerases and transcription factors throughout the nucleus. We can just keep them all clustered right here in certain regions of the nucleus, and then bring the DNA that needs to be transcribed into those regions. Now, before a gene can be transcribed, though, in eukaryotes, some of that chromatin structure, those nucleosomes, have to be removed. So I want to move on into that direction right now, talking about chromatin structure. So let's talk about chromatin remodeling. So let's draw a piece of chromatin here. And I drew 3 regions here that I'm going to wrap up in histones. And we've talked about histones before, but let's just remind you of a few features that are important. These histones, 8 of them altogether. There's 4 on the other side. You just can't see it because I'm not that good of a drawer. But there are a total of 8. And each one of these makes up a nucleosome. So each of these are nucleosomes. Now, I'm going to take a bit of an artistic liberty here just for ease of explaining this concept. But I want you to know in the back of your mind that any given gene is going to span several nucleosomes. So in order to have access to any of these genes, you would need to pull off histones from a whole series of nucleosomes. But for simplicity, what I want to do is say that there is one gene here, which I'll just call gene 1, and there's a gene here, gene 2, and a gene here, gene 3. Just for simplicity in explaining how it is important to remove these histones. And we can discuss this in terms of our example we had earlier, where we talked about insulin and crystalline. So we can say then here, just for our model, that gene 2 is insulin. And we can say gene 1 is crystalline. But remember, these two genes in reality would not be right next to each other, and none of these genes would be within a single nucleosome. So let's say that this DNA here is in a pancreatic cell. And more specifically, we're in a beta cell of the pancreas, just to keep everything legal here. And so in this beta cell of the pancreas, we want to turn on the insulin gene. As it is written now, any transcription factors, and we'll talk in great detail about transcription factors in a moment, but for now just remember that transcription factors are necessary to have access to the gene in order to turn the gene on to ultimately make the protein. But as it's drawn now, transcription factors would be blocked from accessing any one of these genes because they are wrapped up in the histone proteins. So we need to remove these histone proteins in order for the transcription factors to turn on the insulin gene. But we want to keep the histone proteins on these other genes so we don't turn those on. And I think to understand how we're going to remove these histone proteins, we have to think about why they are bound there. They are bound there because DNA, because of those phosphate groups, is collectively very negatively charged. The histone proteins, however, contain positively charged tails, and these positively charged tails allow it to interact with the negatively charged DNA. Simple but strong ionic bonds are forming here. So I'm going to erase the transcription factors for now. We'll come back to them. But there is another set of proteins that I need to talk about that are going to be important in pulling these histones off. The first enzyme I'm going to talk about is an enzyme called histone acetyltransferase. And we will usually just call these HATs. And what histone acetyltransferases will do, as the name implies, is they will take an acetyl group and they will add it to the histones, at the positively charged amino acids of the histones. And by doing that, it neutralizes this positive charge. In neutralizing this positive charge, these histones now will fall off. So let's go ahead and redraw this region here with the histones absent. So I'm going to draw the histones collectively over here with the acetyl groups on them. They have fallen off the DNA. This allows this region that was once protected by the histone proteins to, for a lack of a better way of drawing it or saying it, to unfurl, to fall, to open up. Kind of like a ball of yarn. If you were able to pull out that one piece and release the rest of it, it would just all fall apart. That's kind of what's happening here. And by doing that, transcription factors now have access to this DNA. And we'll talk, as I said before, in great detail about this, but let's say the promoter is right here. Now that we just released the DNA, transcription factors bind to the promoter, recruit RNA polymerase, and now we can turn this insulin gene on. We don't have time to talk about it in this class, but if you take a molecular genetics class, and especially if you take an epigenetics class, we'll learn more precisely how these histone acetyltransferases will selectively add an acetyl group just to this gene, just to the histones at the gene you want to turn on, but not to the histones of the genes you want to keep off. Suffice to say, it's a much more complicated process than I just drew here, but what I want you to know is that these HATs will add acetyl groups to the histones, cause the histones to fall off, releases the DNA, opens it up, so that transcription factors can turn these genes on in a very specific manner. Okay, now, presumably you don't want to make insulin all the time, even if you are a beta pancreatic cell, or if you happen to be talking about a cell that is not a pancreas cell. But let's say we're still in the pancreas, and we want to turn this gene off. that's when another protein comes along. And this other protein is called histone deacetylase. And we often call these HDACs. And what these HDACs do is they'll come along here and remove the acetyl group, as the name might imply, deacetylase. So it removes these acetyl groups. So let's remove these acetyl groups. And now that positive charge on these histones is once again exposed. And now they can associate with this DNA here, where the insulin gene is, and wrap it up again, thereby effectively preventing the transcription factors from having access to it once again. In the next unit of material later on in the semester, we'll talk about cancer. And we'll come back to this and talk about how turning these genes on or off is really important in the progression of cancer. But you'll have to come back later in the semester to hear that story. What I want to move on to next is the idea that once this DNA has been exposed, because the histones have been removed, how are these transcription factors and other proteins going to be important in turning the gene on? Because just because we expose this DNA doesn't mean that it's automatically going to turn on that gene. It takes a precise coordination of proteins like transcription factors to bind to the DNA at the right time to turn those genes on. It's not just an on and off switch. It's a very complicated process as we are learning. Okay, now let's talk more specifically about gene regulation at the specific transcription level. On the past couple podcasts, we talked about how we get that DNA into these transcription factories and then how we remove histones. And the whole purpose of that is to expose the DNA that needs to be transcribed. So that's what we want to talk about now. Now that we have that DNA opened up, how are we going to turn on a specific gene? So let's go ahead and get started. And we're going to start with talking about transcription factors. And we know that transcription factors are very important. And we know they're important not only because we know they turn on genes, but when we compare, let's say, humans with chimpanzees, one thing that we know, and you've probably been told many times, is that approximately 97% of our genes are the same. That always boggles the mind to think about how phenotypically different we are from the chimpanzees, yet genetically we are very similar, extremely similar with the chimpanzees. And they've done a study where they compared which genes are being turned on and off when we compare different tissues between humans and chimpanzees. Without going into great details of this study, what they were able to find is that there's approximately 1,000 genes of about 25,000 genes total. About 1,000 of those genes have a significant difference in expression. These 1,000 genes, they went back and were able to determine that their differences of expression are controlled by 90 different transcription factors. Humans have about 2,000 transcription factors, 2,000 to 3,000 transcription factors, but only 90 of them play a direct role in differentially expressing these 1,000 genes, which largely helps explain the phenotypic difference between humans and chimpanzees, even though we are so genetically similar. So transcription factors, the take-home point, are very important in the individual, but what we're learning is that they're very important in understanding evolution. So now I want to dive into what these transcription factors are doing at the DNA level. And some of this won't be new to you because we've already talked about transcription factors in a general sense, but I want to talk about them in a greater detail now. So now that we can see how important the transcription factors are, I want to draw a gene here, and I'm just going to draw it as a single line, this large piece of DNA here. And in this drawing, I want to explain how a gene can be lightly turned on or fully expressed in eukaryotes. And as you can imagine, it's going to be a little bit more complicated than what we talked about with the lac operon. But some of the basic principles are still there. There's just more players involved, more proteins involved. But at the end of the day, what we're trying to do in both bacteria and eukaryotes is getting that RNA polymerase stably attached to the DNA so we can turn these genes on. So what I'm going to draw here in this picture is how we're going to activate transcription. And we might as well be consistent. Let's talk about our insulin gene. And let's put our insulin gene way down here. The sequence for the insulin gene is right here. And to remind you from a previous podcast, about 25 base pairs upstream of the insulin gene is that core promoter. Remember, we called it the Tata box because it contains that consensus sequence of TAs and TAs. And so at this region here, there's going to be several general transcription factors. Remember, those are the ones we call TF2 for transcription factor 2 because it brings in RNA polymerase 2. And we talked about several different letters. Remember, transcription factor 2D is the one that brings in the Tata box. But in the end, we know there are several of these general transcription factors that bind all over this core promoter. And in doing so, remember, they recruit RNA polymerase. I'm just going to draw RNA polymerase like this over the core promoter and beginning to transcribe the insulin gene. So this is RNA polymerase. And what I didn't tell you about in the previous podcast is that these transcription factors, while they are important in bringing RNA polymerase to this promoter, the ability to transcribe this gene, in this case the insulin gene, is weak. It can still transcribe it, but in a much more, what we might call basal level, that leaky expression. Just enough to make a few transcripts, but not to turn it on full blast. And that's largely because its association with the DNA is not as stable as it would need to be to turn it on at a high level. And so we need other proteins that are going to be involved here to stabilize this interaction. And so I'm going to draw several other proteins here, as well as several other regulatory sequences. There are two main other regulatory sequences I want to talk about. The first one I'm going to put right here, and these are typically a little bit upstream of the core promoter. And we're going to call this a regulatory promoter. And it's going to be, since we're talking about an activation here, it's going to bind something we call a transcriptional, and I'm going to abbreviate this, transcriptional activator. And it's going to help stabilize this RNA polymerase. Now, depending on which gene we're talking about and the level of expression the cell desires, there may be another protein involved here. And this protein we're going to call a co-activator because it interacts with the transcription activator to bind to the RNA polymerase to stabilize it. Now again, depending on the level of control and the degree of expression that is desired, there are sometimes additional regulatory sequences. And so I'm going to draw an additional regulatory sequence up here. And I've intentionally drawn it far away because these sequences, which are called enhancer sequences, are typically found thousands of base pairs away. Sometimes it's hard to imagine how they could help affect this gene. That's, again, thousands of base pairs away. And what was discovered is that sometimes we mistakenly think of the DNA as just this long linear macromolecule. But it's important to remember that it's very dynamic and it loops around quite a lot. And so if it loops around here, anything that binds to this enhancer sequence here can now have close proximity to other parts of the DNA. And that's exactly what happens. To this enhancer sequence, we will have additional transcriptional activators bind to it. Now, usually this transcriptional activator doesn't immediately or directly bind to RNA polymerase, but usually what happens is another protein, because why not? We just keep throwing proteins at this. And this other protein is called a mediator. And often this mediator protein will help stabilize the interactions between this transcriptional activator, these transcriptional activators at the regulatory promoters with the co-activator, help stabilize all those proteins in combination with the RNA polymerase and the general transcription factors, so that this is a very stable complex. And so that once all of these proteins are present, we get a much stronger and much more robust expression of, in this case, the insulin gene. As you can see here, I've only drawn a few proteins. The actual regulation of various genes in eukaryotes involves many more proteins than I've drawn here. Sometimes there are many mediators or many different regulatory proteins. All these proteins together help to precisely turn on a gene at the exact level that needs to be for that cell. It's not an on and off switch. It's much more like your kitchen faucet, where you can turn it completely off or you can turn it completely on, but there's so many other variations in between. And that's what's happening here at eukaryotic genes as they are being regulated. Now, sometimes you want to make sure this gene is turned off, and sometimes you need to turn it off in a very rapid manner. And sometimes you need to make sure it's turned off completely. And to do that, sometimes there will be additional sequences here, far downstream, that we will call silencer sequences. And they will bind various other repressors whose jobs are to disrupt the interactions of these proteins in one way or the other by various mechanisms. But for the purpose of this class, I just want you to know that there are these silencer sequences that bind repressor proteins that prevent this stable interaction. All right, now I want to talk about a problem that potentially exists with enhancers. As I said, they can interact with genes 1,000 base pairs away. But there are also other genes, say off this board up here, that are also 1,000 base pairs away. What's to prevent this enhancer from helping to activate this gene and other genes that are also far away? And so I want to talk about the role of something called insulators. So let's draw a long stretch of DNA, and I'm not going to loop it around here. I'm just wanting to draw a single stretch of DNA here. And I want to draw an enhancer here, and we'll just call it enhancer 1, and I want to draw an enhancer here. And enhancer 1 helps enhance the expression of, let's say, gene 1, and enhancer 2 helps enhance the expression of gene 2. However, as we said on the last whiteboard, DNA is very flexible. And as this chromosome spins around, it's just as likely that this gene 2 could loop around and be in close proximity with enhancer 1. But for the sake of this argument, we want to make sure that enhancer 1 only enhances the expression of gene 1, and enhancer 2 only enhances the expression of gene 2. We don't want cross abilities to enhance the expression of both of these genes. And so what they've discovered is that there are these special sequences here that we will call insulators, insulator sequences. And what these insulator sequences will do, and this is a very clever name, they will bind proteins, or several proteins, which we're going to call insulator binding proteins. These insulator binding proteins, by various mechanisms, prevent enhancer 1 from enhancing the expression of gene 2, and they prevent enhancer 2 from enhancing the expression of gene 1. Okay, that ends this podcast, but I just want to remind you of the few things we talked about, because we talked about a lot of different things in this podcast. Remember, we talked about chromosome territories and chromatin and transcription factories. And this was just the large view of how chromosomes are laid out in the nucleus and how certain areas in the nucleus is where transcription occurs. And then we moved into chromatin remodeling, how we remove those histones in order to turn on a gene and how we add histones to turn off a gene. And then we spent the last part of this podcast talking in great detail about transcription factors and how they can be used to precisely turn a gene on or off. This is a more gross overview, making that DNA available, and then here is that fine-tuning of that expression. How do we get that gene turned on, and at which level is that gene turned on? All right, that's all I have to say in this podcast. In the next podcast, we're going to talk more about, after that RNA is made, how is it modified? How is it used to regulate whether or not that protein will ultimately be made? If you have any questions over this material, please come see me. If not, I'll see you in class. Bye.